Okay. I think we should probably get started. My name is Douglas Hilton. I'm the Director of the Walter and Eliza Hall Institute of Medical Research, and it's my very great pleasure to welcome you all here today. I'd like also to acknowledge the Wurundjeri people, who are the traditional custodians of this land, and I'd like to pay my respects and respects on your behalf to the elders, both past and present, of the Kulin Nation, and extend that respect to other Indigenous Australians present. As I introduce the Institute, I'd like to give you a little bit of a picture of why we're interested in cancer research. We're an organisation that is celebrating our centenary in 5 years' time, and for 55 of our 95-year lifespan to date, we have had a major role in cancer research in Melbourne, nationally and internationally. You might think for a scientific institute that interest in cancer research was because of exciting scientific questions to be answered, or wonderful papers to be published in esoteric scientific journals, or because as scientists we thought that there might be Nobel Prizes, fame and fortune at the end of that journey. What I can tell you is that probably like everybody else in the community, we have family, friends and people we know in the community that have been touched by cancer. We understand from talking to those people, talking to our family and friends, and also from talking to the clinicians who treat cancer patients, many of whom work in this institute. We have a large number of scientists who have clinical appointments in the hospital and also work in our institute. We know from talking to those clinicians that that battle with cancer and their capacity to treat cancer patients is far from satisfactory. We know we have to find better ways to battle cancer for two reasons. One is that those patients that battle and survive, most of us know, go through horrendous changes in lifestyle because of the side effects of that chemotherapy. So one of the things that we need to do better is to find better tolerated therapies for those cancers that we can already treat. And the second reason, obviously the most tragic reason, is that not everybody gets through that battle. So despite the 55 years of cancer research, we know that there's a lot of work, additional work to do, and that's why, as you'll, I hope, see today from what Andrew says and from the opportunity that you might have talking to our cancer researchers after the lecture and the panel discussion, that's why we've got such a passionate group of researchers and clinicians that are working together to try to do better for the people in the community, our family, friends, and no doubt all of you who have suffered that fate and have battled cancer as well. We are in the middle of a major building program, so you might have seen as you walked in the institute that it's more like a construction site than a laboratory. That's the front end of that building program, but at the back end we have a new building that will double the size of our institute and will be open towards the end of this year. I'm really delighted to be able to announce that cancer research will remain an absolutely major focus of this institute as we expand. In fact, as we move into the new building, we'll be able to devote nearly 3 times as much laboratory space and effort to cancer research as we have in the past. But as you would also know, buildings don't make discoveries. We need the best scientists, and those scientists to be well equipped and to be able to take a long-term perspective in the research they do in order for us to be able to make the breakthroughs in the future that we've also made in the past. As an institute, we're very proud of the sort of discoveries that Don Metcalf has made about colony-stimulating factors, discoveries that have led to better treatments for more than 10 million cancer patients worldwide. We're also very excited about discoveries that were made nearly 20 years ago to try and understand better how cell death is controlled and how cancers evade that cell death process, and which over the last 20 years have now led to clinical trials that I think Andrew may mention that are going on at the Royal Melbourne Hospital and that we think will yield a new dawn for treatments of many different blood cell cancers. So we're excited about those discoveries. We're proud of the discoveries we've made in the past, And we know those discoveries can only be made by well-supported, talented individuals. And it's a real delight to see a number of representatives from the philanthropic organisations that have provided the long-term support for people like Don and Andrew. And they include Cancer Council of Victoria, who supported Don for 55 years with the Carden Fellowship. David, it's great to see you here. I know we've also got members, staff from the Leukemia Foundation that support a large number of our researchers across a number of divisions. And if you have the opportunity, perhaps at some other time, to take a tour of our laboratory, you'll see equipment there that has been provided by the Australian Cancer Research Foundation, the ACRF. We've needed the support of the Cancer Council of Victoria, the Leukemia Foundation and the ACRF, along with many other donors and supporters and individuals within the community to allow us to make the discoveries that we know have changed the lives of many Victorians and Australians that have suffered cancer. As I said, we're nearly tripling the size of our cancer research effort in the future, and I'm sure that that increase in effort will lead to increased benefits for cancer treatment. But you can take it for granted that in order to be able to make the most of the talented scientists, we need also to triple the support that we receive from the community. We know we're going to be very competitive when we go in for NHMRC grants through taxpayer-funded organisations, but we also rely very heavily on the day-to-day support that we receive from members of the community and organisations like the Cancer Council of Victoria, the ACRF and the Leukemia Foundation. So we're really excited about the future. We think there are great experiments that are going on now that will lead to changes in the way cancer is treated. We hope that your visit here won't be your first visit. One of the things that we're passionate about is having the community walk the journey with us as we undertake research. We would be delighted if you would keep up to date with our activities and share some of our triumphs and obviously some of our disappointments as things don't go so well as we hoped. We hope you'll be part of that journey through reading magazines like Illuminate and that we send out to our supporters, but also perhaps coming back and being part of a discovery tour where we have smaller groups of the community come in, visit some of the laboratories and understand in a little more depth what's going on. I do have a couple of housekeeping announcements before I introduce Andrew, and that is just to say that this event is being filmed and will be available on our website or via DVD. So if anybody's interested in following up or passing on the proceedings of these events to friends who could perhaps not be here tonight, that's a real mechanism for you to be able to do that. And without further ado, I'd like to introduce Dr. Andrew Roberts. Andrew is one of those people that I mentioned. He spends 50% of his time in the Royal Melbourne Hospital, where he is a clinical haematologist and a bone marrow transplant physician within the Department of Clinical Haematology and Medical Oncology. In this institute, he's the head of clinical translation, an appointment that he recently received, and is a laboratory head in the division that was started by Don Metcalf and is now run by Nick Nicola and Warren Alexander. That is the Division of Cancer and Haematology. Andrew, it's a pleasure to introduce you, and we're looking forward to your talk.
Doug, can I get this?
You can have this, otherwise you'll be able to hear what I say to Deb. It's all on.
Thanks very much. Well, thanks for that introduction, Doug, and welcome to everyone here tonight. Thanks for coming out in the cold. I hope to provide you with a little bit of an update with a slightly biased perspective about the role that research plays in improving treatment for patients with leukemia, lymphoma, myeloma and other cancers of the blood. And following my talk, we hope to be able to answer questions that you may have, and I'll have assistance with that through a panel of both seasoned and younger researchers who are making it their life's work to try to come up with ways to improve the lot of patients who are diagnosed with cancers of the blood. So the emphasis tonight is really on where research sits, what role it can play, the differences it's made already, and I'd like to give you an example of where I think it will be making a difference in the next few years and the decades beyond. First off, though, it's worth setting the scene. You wouldn't be here tonight if you didn't already have a personal perspective on the impact leukemia, lymphoma, myeloma, myelodysplasia, or other serious blood disorders have on people in our community. But it's worth just reflecting on just how big a problem it is in Australia today. The figures here from 2005 suggest that there were 9,700 new diagnoses of people with cancer of the blood in 2005. Given that the rate of diagnosis is increasing by several hundred per year, that means here in 2010, over 10,000 people will be diagnosed in 2010, and that number will continue to increase over the next decade at least. Once people are diagnosed, they will typically receive treatment, and currently there are over 50,000 Australians who have been diagnosed, perhaps treated, and maybe survivors of blood cancers at any one time. And again, that number of people will be increasing both because of increased diagnosis and also because of increasingly successful treatment. There are some very major challenges ahead of us when we think about blood cancers in the Australian community. As I've already alluded, the rate of diagnosis is increasing faster than the rate of growth in our population. And that means that we will be seeing more and more patients in our clinics, in our hospitals, presenting with blood cancers. Sadly, many thousands of people diagnosed with blood cancers die each year, and clearly cure is not possible for many patients with these diseases. Because blood cancers can affect us from very early in life through our teenage years, our middle years, our senior years, and also in the very latter stages of our life, many years of productive life are lost when we are unable to cure cancers of the blood. And so that the figures here of over 20,000 productive years lost in the Australian community every year obviously far exceed the number of patients diagnosed and dying. So there are major challenges that we must meet, both from a community perspective and also from the very personal perspective of people we know and love and meet who have cancer. Now, research plays an integral role in all the improvements in medicine over the last 150 years. And basically, when we think about cancer research, I think it can be broken up into four broad areas. I'll just background you there. The sort of work we principally do here at the Walter and Eliza Hall Institute and at a number of other institutes around Melbourne and around the world is try to understand the biology of cancer. That's the nature of the disease, what causes it, where does it come from, what are the changes in cells that lead them to grow outside the body's control. And as you've heard from Doug, this institute's been involved in that type of research for the last 55 years since the arrival of Professor Don Metcalf here with the assistance of the Cancer Council Carden Fellowship. And in all those years, there's been a heavy emphasis on cancers of the blood. As well as trying to understand the cancer itself, we need to understand how cancer and patients with cancer are made up in our community. And this type of work looks at population-based aspects of cancer and is really the way which we can identify how common cancer is, what types of blood cancer are increasing and decreasing, It's the sort of research that's needed to be done to identify the cause of cancer, particularly if it's related to an environmental agent or a lifestyle agent. And so this type of research is crucial if we're trying to work out whether blood cancers are increasing or decreasing and what the cause is. Once that's been identified, we can then think about, are there ways to prevent blood cancers from occurring? And again, public health research tells us the best way to move into prevention. Clinical trials, we're all familiar with those. That's the way we systematically evaluate whether a new treatment idea or a new strategy for improving outcomes for patients works. It relies particularly on the involvement of people who are affected with leukemia, lymphoma, myeloma themselves, as well as the doctors and nurses in the hospitals and in the clinics and the carers for patients. And finally, there's an area of research which I won't be touching on much tonight, called health services research, and that's really a relatively new field of research that tries to understand how we can best integrate all the knowledge from these areas into the delivery of the best possible care for patients who have the disease today. Now, I'm not going to dwell on that area of research much, because I can tell you from my own experience that in fact for leukemia, lymphoma, myeloma and other cancers of the blood, there is very rapid adoption of new treatments, new knowledge and new ways of caring for people currently with the diseases, particularly through clinical trials. And so for my patients with blood cancer, we don't need much research at the moment into how to best deliver the new treatments. What we're desperately in need of is new knowledge coming out of these areas of research. Now, in the advertisement for this lecture, we said that we'd be identifying what the current big questions in blood cancer research are. And unashamedly, this is a personal view of mine, grown out of 25 years of experience in treating patients with cancers of the blood, and also the last 20 years of researching how to do that better. But I think you'll find that many people around the world would tend to agree that these are the really big questions. And how do you define a big question? Well, I think the way to think about it is, if we had the answer to these questions, would that make a big change into the outcome for people diagnosed with the disease? And as we go through them, I think you'll see why I think these are critical questions. Firstly, what are the causes of these cancers? I will touch in some detail on this in the moment, but it's fair to say we don't have a complete handle on what the causes are, and that has implications particularly for the second question, which is, can we prevent some or all cancers of the blood? I'm sure you're all very aware of major programs of prevention for some cancers in the community. Cancer of the cervix can now be prevented through early detection programs of pap smear, and in fact it can now be prevented by immunisation against the human papilloma virus. And so there's a cancer that is becoming less and less a problem for the Australian community, because we're able to prevent it from developing. Similarly, we're able to also prevent cancer of the lung and some other cancers by ceasing smoking or preventing people from taking up smoking. Unfortunately, as I'll touch on a little later, we really don't have a great idea about how to prevent cancers of the blood. And so for me, that's a big question. It's not one I'm going to be able to answer for you tonight or in the near future. The next three are linked and are really going to end up being the main focus of my lecture tonight. Really focusing on trying to do better in the way that we treat people once they have developed cancers. Firstly, we want treatments that when they're first used are more effective in reducing the burden of cancer in the patient, perhaps reducing it to nil. Once we've successfully controlled or hopefully eliminated the cancer, the next challenge is to prevent it from recurring. And we certainly need further research into understanding how that can occur. And as Doug talked about at the beginning, even in those blood cancers where we now have what we would consider successful treatments, there's a major impost on patients going through those treatments in terms of side effects, what's called toxicity to doctors, and it would be wonderful if we can reduce these. If we turn to the first big question, what are the causes of these diseases? Well, from basic research done in this building as round as other institutions in Australia and around the world, we now have a pretty good understanding that they seem to arise not because of one single event in a person's lifetime or because of a gene that they've inherited from their parents, but because of the accumulation of a damage in a number of genes, the genetic code, in certain blood cells. And typically this occurs during life, as they grow through life. Only rarely is this inherited. So most blood cancers are diseases that we develop as individuals as we go through life. There are some environmental factors that do induce cancers of the blood. We know that high-dose radiation causes cancers of the blood. We know that from the nuclear bombs dropped on Japan, as well as the exposure of patients at the Chernobyl nuclear disaster. We know that high concentration of some chemicals, such as benzene, can cause acute myeloid leukemia, and hence this has been largely removed from workplaces. We know that chemotherapy for other cancers can increase the risk of developing leukemia at a later stage for people who've been cured of their original cancer. We know that some virus infections in some circumstances can lead to leukemia and lymphoma, and collectively all of these increase the risk of developing these diseases. We also know that if we suppress our immune system, and that often occurs in situations of medical treatment or following tissue transplantation, that there's an increased risk of cancers, particularly of the blood, and that's because the immune system, as part of its job, is there to survey our body and try to rid us of any blood cancers that are beginning to happen. Despite this large amount of knowledge which has built up over the last 30 years, for most blood cancers we haven't been able to identify a specific cause, and indeed lifestyle factors that we associate with being major cause of other cancers such as cigarettes, alcohol and obesity may play a small role for some blood cancers, but not to the same extent they do for some diseases such as lung cancer, cancer of the liver and bowel cancer. And so these are open questions, they're under active investigation. I'm not sure when the breakthroughs will occur, but when they do, this will give us major new insights into better ways to look after people with this disease. The great goal, of course, would be able to prevent them. Currently, we don't have any good strategies for being able to prevent cancers of the blood. The reason for that is we simply don't have the knowledge about what we would need to do to prevent that. Unlike some other cancers, early detection of cancers of the blood also doesn't appear to be a major advantage to people. I think you're all aware that for common cancers like cancer of the breast and cancer of the bowel, that the outcome for people diagnosed with these diseases is often much better if the cancer's caught early. We know from research over the last 20 years that typically catching a cancer of the blood early is not a major advantage. It's not a disadvantage to people, but it doesn't seem to lead to the same sort of advantages we see in some other cancers. And again, this is a reason why there's no push in the community to go out there and try to get these cancers early, because it's of no benefit to patients. Despite what we don't know, we need to keep doing this type of research because, as I said earlier, the incidence of cancers of the blood is rising at a faster rate than the growth in our community, and the risk is rising, particularly as our population gets older. And also at any age, young, middle-aged, elderly, the risk for people in our community developing two blood cancers, non-Hodgkin lymphoma and myeloma, is increasing over the last few decades. And I'll just give you an example of this for NHL, or non-Hodgkin lymphoma. This is a graph, red for women, blue for men, and for many blood cancers it's more common in men than women, again for reasons we don't understand. And this is the number of diagnoses per 100,000 members of the community. It's over time from 1980 through to just near the present day. And if we look at men, back at about 1982, 15 diagnoses were made for every 100,000 males in our community. That has now gone up to 23 per 100,000. And this is adjusted for various risks at any age. So this is a very marked increase in the rate at which this is being diagnosed. Similarly for women, although not quite as great an increase. The other thing to note is that most blood cancers get more commonly diagnosed the older we are. So blood cancers are a disease of ageing, and our population is generally ageing. And again, when you're young, the risk of developing lymphoma is very low. If you're 80, your risk of developing lymphoma is now about 1 in 1,000 per year, which is over 10 times higher than it was, 100 times higher than it was when you were a baby. The only exceptions to this in the blood cancers are acute lymphoblastic leukemia, which is far more common in children, uncommon in adulthood until senior age, and Hodgkin lymphoma, which is also common in adolescence. But for non-Hodgkin lymphoma, these two factors mean that the number of patients diagnosed with non-Hodgkin lymphoma in our community is rapidly rising. Here's 1980, about 800 diagnoses per year in Australia in men. They're in the blue, red for the ladies. 2005, 3 times as many people diagnosed. And that's in under 30 years. So rapidly rising, becoming more common disease. And this is one of the most rapidly rising cancers in the community. Another one that's paralleling this is multiple myeloma. And so 2 common blood cancers are becoming more common. Given that we can't prevent them, given that we don't really understand what it is about our lifestyle and how we live in Australia and the Western world that leads to the development of cancers, we really need better treatments for the people who are presenting with cancers now and in the next few years. We have to be able to more effectively treat them once the cancer's present. We certainly have to be able to develop treatments that will stop the disease coming back after the initial treatment. And for some diseases where the treatment chances of cure are very high, we want to be able to reduce the side effects of treatment to minimise the burden that they have for survivors of blood cancers. And the theme for tonight really is that the likely keys that we're going to be able to do that is by identifying the Achilles heel of each type of blood cancer and try to target the weakness of these blood cancers through new therapies to overcome resistance to the current chemotherapy radiation treatments that we use today. Now, one question that I get asked quite a bit is people are all very enthusiastic for research, but is there actually any evidence that it's made a difference in the last 10, 20 or 30 years to how we look after people with blood cancers? And more importantly, has it improved the chances of being cured? Has it improved their quality of life? Has it made it easier for them? And I'd like to give you 4 quick examples of evidence that tells us that the answer is an unequivocally yes, and furthermore gives us the way forward to do more and more of this type of work. The first example is outside my area of expertise. It's caring for children with cancer, acute lymphoblastic leukemia. But the story is very well known. In the 1950s, if your child was diagnosed with acute lymphoblastic leukemia, there was no treatment. You were advised to take your child home, not bring them to hospital, because that would just distress them. There was nothing that could be done. You would nurse them at home and try to treasure the last few weeks, days and months of their life. In 2010, approximately 80% of all kids diagnosed with this terrible disease are now cured. And so the challenge for those in that respect is now to try to make the treatment less toxic, less burdensome on children and their families. The flip side, of course, is for the 20% of children who aren't cured of the current treatment that we desperately need new treatments. If it can be done for 80%, we should be able to do better. We should be able to get that to 100%. If in 50 years it's gone from 0% to 80% cure, surely we can go the extra 20%. If we return to the non-Hodgkin lymphoma I was talking about, I want to show you some further Australian figures that tell us unequivocally that we're getting much better at treating this disease. So non-Hodgkin lymphoma, again, this is the rate of diagnosis, rate of death now from non-Hodgkin lymphoma per 100,000. Men in blue, red in yellow. Again, going back to the 70s here, and you can see the rate of death rapidly rising, and that's because it was becoming more and more common, not because we were getting worse at treating it, just it was getting more common. So more Australians dying of the disease, and then somewhere around here, a dramatic fall off. And what's more, the rate of decline is increasing, so that in 2010, it's down here, and we think it may well be falling. If we turn that into total number of patients dying, so that's the rate per 100,000 people, this is now the total number of deaths in Australia per year, and you'll remember that we have to now account for the fact that compared to here, there are 3 times as many people diagnosed with non-Hodgkin lymphoma now as there was back here. Again, the rate of death rapidly rising, and now, even though the numbers of patients going up, it's turning down, and beginning to follow this pattern. Why has that happened? It's simply happened because of the introduction of a new drug that has complemented the old chemotherapy drugs that we'd used for the previous 20, 30 years. That drug is something called rituximab. The details are not important, but basically it was a new style of treatment designed to target some types of non-Hodgkin lymphoma and using antibody technology that hadn't previously been used for cancers. This has become now very common. It's a blockbuster drug for the pharmaceutical company that developed it, but I think you'll agree that any investment is worthwhile for what it's been able to do with people diagnosed with non-Hodgkin lymphoma. Now, the number of deaths from non-Hodgkin lymphoma has dramatically reduced. As yet, we haven't necessarily turned all those reductions in death into cures, and so there's still plenty of work to be done with non-Hodgkin lymphoma. But this is very clear evidence of the effect that one breakthrough that's taken through to development of a new treatment can have for many, many people. Similarly, there have been recent advances for patients with multiple myeloma. This is a disease I treat many patients with multiple myeloma, and the outcome for people under the age of 60 improved dramatically in the late 1990s through the introduction of a treatment called autologous stem cell transplant, and I'll touch on that a little bit later because it's something that became readily available through some breakthrough work done by Professor Donald Metcalf here. But for people over the age of 60, there'd really been no improvement in treatment for last 25 years of last century, until along came 3 new drugs, the first in 1999 and the last 3 or 4 years ago, thalidomide, bortezomib and lenalidomide. And now this is changing the landscape of treatment for patients both young and old with multiple myeloma. None of these drugs yet appear to be a cure, but clinical trials are underway trying to develop the first curative treatment for this very tough disease. And it's now very clear, as the clinical trials are being reported, that the survival rate for patients entering studies in the last few years is now many years longer than the survival rate was for patients entering studies a decade earlier. And I don't have the figures to show you, to prove to you that I do for NHL, but I'm very confident that within a few years we'll be able to show you the same sort of figures for multiple myeloma. Again, so research into developing these new drugs has made a difference. And perhaps the best evidence of all is for one of the relatively uncommon cancers of the blood called chronic myeloid leukemia. Approximately 300 patients diagnosed every year in Australia. This is a relatively slow-burning cancer that inevitably used to develop into an untreatable acute leukemia that rapidly led to people's death. We've known through 30 years of research that it was caused by a very specific change in the genes. But in 1998, despite knowing all of that, if I was seeing a patient newly diagnosed with chronic myeloid leukemia, I would be saying to them, you have an 85% chance of passing away from this disease within the next decade. The only exception to that were patients who were able to undergo a bone marrow transplant or a stem cell transplant from either a brother or sister or another donor. However, 12 years later, seeing the person in the same situation, I can now tell them that they have a greater than 90% chance of being alive in 10 years from now, and that I have no idea what they're going to die of. It may be old age. And the reason for that is the introduction of a new drug called imatinib is the scientific name, Gleevec is the trade name, and basically this is a drug that when taken turns off the switch within the chronic myeloid leukemia cells that drives them to be cancerous. It is the culmination of research into the biology of chronic myeloid leukemia that began back in the early 1960s. And following on its success, there's been a rash of other drugs that are doing similar things in this disease and in another of other cancers that are similarly driven by one major genetic change. And so, as well as the good news for patients diagnosed with chronic myeloid leukemia now, it also means that we do very few bone marrow transplants for chronic myeloid leukemia now at the Royal Melbourne Hospital, whereas it was our most common indication just over 12 years ago. So new research leads new advances to treatment, changes old treatment, and allows us to get rid of some of the more toxic treatments. So I hope you can see from the theme of where I'm going that I think the biggest advances in treatment for patients with blood cell cancers in the next decade or two is likely to come through the nexus between studying the biology of cancer, development of new treatment, most likely drugs, testing them in clinical trials, and then taking knowledge back and forward between these processes in an iterative process to find out the best way to develop the new drugs that work best to make difference for patients. And the central idea is to try to identify a central or sentinel abnormality of the blood cancer cells. And this will differ from different type of blood cancers, and this is potentially a barrier to our advance. Once this abnormality has been found, to target this abnormality or Achilles heel, and this may mean the development, or usually will mean the development of new drugs specifically purpose-made to do this. And then once they've been made, we need to take them in clinical trials to see how effective the new strategy is. If it's not perfect, we have to go back to drug development, might have to go back to cancer biology, and come back with better drugs for new clinical trials. Now, I want to give you an example of how Australian research has improved patient care for people diagnosed with blood cancers. And I want to tell you this story for several reasons. Most importantly, to give you the message that firstly, this process that I've been talking about takes a very long time. As you heard from Doug, Professor Metcalf began researching here at WEHI in 1955 with the support of the Cancer Council of Victoria, and that was in cancer research. At that time, there was very little cancer research done. So one message is it takes a long time for discoveries to turn into new treatment for patients. The other message I want you to take away is that this is work that may be led by an individual such as Professor Metcalf, but typically is the product of teams of individuals, many of whom are just as talented as the leader themselves, and that they come together to work for the benefit of patients with these diseases. Now, Professor Metcalf started his work in 1955, but it was really in the mid-60s where he began this work, and which I'll tell you about a little more in a moment. Here are some of the highlights. He really opened up a whole new area of research into blood cancers. His discoveries directly led to 2 new drugs that are now, I prescribe, one of them at least, G-CSF, every day and have done for the last 10 years. It's now just part of what we do. We take it for granted in clinical medicine, yet it is many years in the discovery. As Doug pointed out, over 10 million patients with cancer around the world have benefited from treatment with these drugs that Don discovered. And within that, that means that probably several 100,000 people have been cured where they could not have been cured before if it had not been for this drug. And it certainly changed forever the care of some patients with cancer of the blood, particularly those requiring stem cell transplantation. Here's the timeline for you. Starting in the 1960s, Don asked what was a big question in cancer research back then. Why and how do leukemia cells grow? We had no idea then. Now we have a very good understanding of that, but by attempting to answer this unanswerable question back in the 1960s, he was able to discover 2 hormones the body makes, which drive blood cell production and turn out to be very effective in driving normal blood cells to help us recover from chemotherapy. That was all discovered with his team, including Professor Nick Nicola in 1983. Then, through clinical trials, through the late 1980s into early 1990s, we learned how to use this to make chemotherapy far more tolerable for patients and reduce the chances of having infection. And then, through further research done at Weehai and in Melbourne, we were able to use this knowledge to revolutionise how we do bone marrow transplants. It was a combination of great science, large teams working together, both in academia and industry, and great support from donors and philanthropic organisations like the Cancer Council. I just want to take you through this nexus between basic research, clinical trials, and care for patients. Here's Don, and he discovered these molecules that led to the growth of normal blood cells, and this was in his quest for trying to develop new therapies, leukemia. These were the sort of scientific assays he was doing in the laboratory. I won't go into the details then, but they led to a drug company called Amgen developing the drug, G-CSF, and then injecting into patients, and this occurred at the Royal Melbourne Hospital, amongst others, as the first sites. Here's a picture of Professor Nicola, at that time a young scientist working with Professor Metcalf, and now the assistant director of this institute, one of the first patients receiving a cytokine injection in the Royal Melbourne Hospital, one of our clinical colleagues here. And within a few years, this led to the routine use of this drug in patients receiving intensive chemotherapy. During the clinical trials, another scientist working with Professor Metcalf in the lab, Uli Dursun, discovered that in fact these hormones, when injected into people, did more than just accelerate the recovery of blood cells after chemotherapy. They also led to the movement of stem cells that would create all the cells in the blood, moving from the bone marrow where they normally live into the blood where they could be collected. Capitalising on this, colleagues of mine at the Royal Melbourne Hospital then went on to test whether we could collect those cells from the blood and use them instead of bone marrow cells to do bone marrow transplants. And the long and short of it is that that was highly successful, and now, by and large, most of the bone marrow transplants we do aren't sourced from the bone marrow itself, but are from the blood after patients or donors receive doses of G-CSF to move the stem cells from the bone marrow into the blood. And the impact of this has been to improve the safety and also the access of patients to transplants. It certainly costs a whole lot less than it used to, and it's improved the curability of leukemia and lymphoma for some patients who have bad disease that would otherwise be incurable with current treatments alone. And just to personalise a little of this for you, when I first came to Melbourne at the end of my specialist training in 1992, this was the typical picture of what it looked like undergoing a bone marrow transplant. Most patients were young. The average age, and this is an adult hospital, was 31. We had no patients over 45 years of age. You're in hospital for, on average, 5 weeks and maybe up to 5 months. And here's the thing that's pretty horrific, is you only had these treatments if this was your only chance, because the treatment itself had a 1 in 4 chance of leading to you dying. Pretty horrific stuff. Just over a decade later, here's a more recent patient of mine in their mid-50s. The average age is now 44, and we do transplants in patients up to the age of 60. It's still pretty tough treatment. You're in hospital for up to 3 weeks, but often you're sitting out of bed not looking all that crook. You've still got a nasty risk coming in, so again, it's not a treatment you have unless there's a very good chance of a good outcome if you survive, but that risk of death is about a quarter of what it was just over a decade earlier. So here's an Australian-grown discovery developed by a US pharmaceutical company that really has changed the way most patients are treated with blood cancers today. And as clinicians, we take this as for granted. But Don, many people are in your debt for your discoveries. So let's come to the future, or to the present, and look to the future. What are the current research questions that we should be focusing on, in my opinion? And they're the ones that I think match the urgent needs of our patients. And by urgent, I'd love to mean relevant to someone diagnosed today, but perhaps I'm thinking more of people in the next few years. We certainly need to develop new drugs to overcome resistance to existing treatments. Even those whiz-bang drugs that I've talked about that have changed non-Hodgkin lymphoma and CML, not all patients respond to them. Even young children with acute lymphoblastic leukemia, up to 20% of people still need far better treatments than we've got. And what we've learned from the biology of blood cancers over the last 30 years is that for a disease that has one name, like acute myeloid leukemia, in fact, there are maybe 10 or 12 different types of diseases hiding within that one name. And what we have to work out for each of these is to best tailor current and future treatments to the specific subtypes of these blood cancers. And we may need to develop new therapies for each and every one subtype. Just to give you a little more perspective on that, for some lymphomas, in some categorisations, there's over 30 different types of non-Hodgkin lymphoma. Now, I think most of them can be dealt with in several new developments, but it may be that we need to develop many new treatments for an increasingly smaller fraction of patients with these blood cancers. And so, returning to what I think is the current nexus, the engine room, if you like, of near future improvements in treatment for patients, it's applying what we know about cancer biology and refining it, allowing us to develop new drugs that will overcome some of the problems of previous drugs, testing them in clinical trials, and always having a to and fro, back and forward between these to refine what we do. I'd now like to move on to a very current example of the application of some basic research begun over 20 years ago at the Walter and Eliza Hall Institute that is now beginning to show real promise in early phase clinical trials. Now, 20 years ago, a key question was, how are these genes that we kept turning up in some blood cancers, how were they causing the cancer? We had no idea. One type of non-Hodgkin lymphoma is follicular lymphoma, and it's associated with particular change in the chromosome structure within the lymphoma cells, not within the normal cells of the person, but within the lymphoma cells. I won't go into the details, but basically that led to the discovery of this gene called BCL2, which is a gene that encodes for a protein in cells called BCL2. The function of that was unknown, yet just about everyone with follicular lymphoma had this genetic change. So it was assumed it had to be very important. The discovery of that translocation was 1984. 4 years later, it was still unknown what this BCL2 was doing. But a young PhD student in the institute, now Professor David Vo, was the person to discover the function of BCL2. At the time, he was a PhD student in the laboratory of Professor Jerry Adams, Professor Suzanne Corey, immediate past director of the Walter and Eliza Hall Institute. And David basically discovered that if there was too much BCL2 in cells as a result of this genetic change, then instead of dying like these normal cells would do if left alone or given a death signal like chemotherapy, they would stay alive. These were cells that didn't want to die even when they should. And this was the first example of any gene that caused cancer by enhancing cells to survive. Because it was the first, it took many years and the work of other people, including Andreas Strasser and a large number of other young scientists working with these individuals. This is Professor Corey, Professor Adams, to work out how stopping cells from dying would lead to the development of blood cancers. And to try to summarise it for you, basically, whenever a cell undergoes bad damage to its genes, under normal circumstances, that cell will recognise that it's fatally flawed and will begin a process of cell suicide so that it dies. And that's a safety mechanism in our body to prevent cancer. When there's too much BCL2 around, it prevents these cells from committing suicide, so they build up in the body and allows an expansion of precancerous cells as well as cancerous cells to develop. And this is the basis of the development of cancer. Now, this is a complex slide, and I'll just simply summarise it for you. That's saying too much BCL2 in a cell leads to its resistance to conventional anti-cancer treatment. And we know that from experiments done in the United States at Cold Spring Harbor earlier this decade, and recapitulated by a PhD student of ours, Andrew Way, and first tested in animal models of lymphoma. And in this animal model of lymphoma, if you don't give chemotherapy to the animal, the lymphoma grows rapidly and the mouse has to be euthanized to prevent it from suffering within about 25 days. However, if you give a single dose of chemotherapy, you're able to cure about 85% of these lymphomas. So this is an example of a lymphoma in an animal, not a human, but that models one of the diseases that we treat in human beings called Burkitt lymphoma, that's sensitive to chemotherapy. Now, if in that same lymphoma you put too much BCL2, no longer does that chemotherapy work and all the mice die. And we've gone on to show that that's also true in humans, and that BCL2 is therefore a very attractive target to try to attack in cancer cells, because its presence promotes the formation of the cancer and also blunts the action of our currently available therapies. Now, people have been trying for over the last 15 years to try to achieve this, and particularly the blood cancers, this is relevant because here's the chance of having too much BCL2 if you have follicular lymphoma. It's about 90%. If you have chronic lymphocytic leukemia, it's about 85%. If you have some of these diseases, which we would generally expect to be able to cure 80 or 90% of people, generally it's not present. And in Diffuse large B-cell lymphoma, one of the aggressive lymphomas. It's present in about 40 to 50% of patients, and those people typically don't do as well on treatment. These two diseases respond well to treatment generally, but are generally considered not curable with conventional treatment, and in part that may relate to the presence of BCL2. Now, in order to develop good treatments targeting BCL2, a lot of work had to be done. Some of that was done by teams such as these individuals at WEHI, now involving my colleague David Wang, who's been a collaborator with me in this area for the last 7 years. And basically, research done in this institute discovered the rules about how we might go about testing whether a drug that was developed to target BCL2 was working and how we would go about testing it. Fortunately, Abbott Laboratories, a pharmaceutical company in the United States, were using science, able to cleverly design a drug which they thought would bind to BCL2 and inhibit its function. Through a lot of work done here, as well as in the United States, we were able to confirm that that's exactly what it did, and that allowed us to then move on to tests or phase 1 clinical trials in patients with blood cancers to see whether this new drug that targeted BCL2 would allow these cancer cells that refused to die when they were treated. And this is the team at the WEHI involved in the clinical trials. Kylie Mason, who you'll meet a little later, Dr. Simon Seong Kaur, who's a paediatric oncologist, and David Wang again. And these are individuals just representing a much larger team. And the work that we've done, supported by the Leukemia Foundation, the ACRF, and the Cancer Council of Victoria, as well as the drug companies involved. Now, I just want to introduce you to a moment to the disease chronic lymphocytic leukemia. It's the commonest leukemia in our community. Importantly, not everyone needs treatment if they're diagnosed with chronic lymphocytic leukemia. About half people ultimately will need some treatment, and about a third of all people with chronic lymphocytic leukemia will die prematurely because of their disease. The converse of that, of course, is that 2/3 will die of other causes, and hopefully not prematurely. Every cell in chronic lymphocytic leukemia, and these are the chronic lymphocytic leukemia cells here, the blue ones amongst the red cells in the blood, have too much BCL2, and they accumulate because of the presence of this drug. Some patients have had multiple treatments and it's no longer working, and they're the people that entered our clinical trials at the Royal Melbourne Hospital and the Peter MacCallum to see whether this drug worked. And we were able to see very quickly that it could work in some patients. Here's a picture of the blood. These are red cells. Here's some normal white blood cells. Here they look perfectly happy. And this is a patient who's just received the first few doses of drug. And here's a CLL cell, and you'll need to take it from me. This is a very unhappy cell that is undergoing cell suicide and will soon be disposed of in the body. Patients with CLL typically have very high white cell counts in their blood, and we saw these drop within days of beginning treatment with these drugs. Now, CLL also causes enlargement of lymph glands, and here's a CT scan, which is a cross-section through the abdomen of a patient, if you imagine being cut across here and then lying down. Here's the kidneys, this is the back, this is the front, and there's this large mass of lymph glands about the size of an Auskick football. This patient had had 7 different types of chemotherapy, and nothing was working. Yet after 21 weeks on this new drug, this is now essentially returned to normal. All this is the tissue that should be here. The footy has gone. Now, I don't want to overstate the results. These are very early phase clinical trials. What we're seeing here is the drug's doing what it was designed to do. It's showing effectiveness in patients where chemotherapy no longer worked. It's causing a shrinking of the chronic lymphocytic leukemia. We have not yet seen someone clear all their chronic lymphocytic leukemia from their system. and the trials are ongoing. It does not work in every patient. It seems to have a major benefit in about 2/3 of people, but there are still 1/3 of people where there's plenty of room for improvement. And we're only at the beginning of the development of this drug. I think it'll work much better in combination with chemotherapy than on its own, but we're beginning those clinical trials now to see where we're going. But I think this is an example, again, of some Australian-based research, again, taken overseas and developed by overseas pharmaceutical company, but coming back to Australia and being of advantage at this stage to a small number of Australian patients, but perhaps in the future it'll make a big difference for some of the people who are most needy for new treatments. And so we're doing clinical trials with this drug in CLL, some non-Hodgkin lymphoma, and in the future, almost certainly it will be tested in paediatric ALL as well as some other blood cancers. Now, because it's not a home run, we're in the business of trying to do better. And so we're going back into the laboratory, expand the team. Now I've got drug designers like Peter Coleman, as well as a whole host of really talented young and old scientists trying to refine these drugs, make them better, make them work more effectively. And we would love to take them back into the Royal Melbourne and Peter MacCallum and do further phase 1 trials in patients who have a desperate need at the present time. And again, this only happens through the support of philanthropic groups as well as pharmaceutical companies. Now, I'm just about at the end of my lecture. I've only focused on one aspect of blood cancer research at WEHI, and WEHI is not the only place in Australia doing blood cancer research, but I think you'll appreciate the time forbids going through everything that's happening in the scene. I said to you earlier that I thought there was a second major current priority area of research, and that's try to understand how to best tailor current and future treatments to specific types of blood cancers. We're going to need younger people than me to get stuck into this one because I think this is really crucial. Ashley's one of the people on the panel. Stefan is not on the panel, but he's an example of the type of researcher here at WEHI who's ready to take on this challenge in other cancers of blood, acute myeloid leukemia, some myeloproliferative diseases, multiple myeloma, and the increasingly recognised disease, myelodysplasia. I think we're going to need advances in different aspects for all of these diseases, and we're going to require an ever-increasing research effort for at least the next 10 or 15 years before I think we can see all the curves for death rates for blood cancers decreasing in Australia. So in summary, blood cancers are an increasing problem for the Australian community, and for those of us with friends, family, or loved ones with the disease, we just know how devastating they can be. Unfortunately, prevention is not currently possible, and we really need some breakthroughs there in terms of our understanding of causation before we will be able to even think about ways of preventing these diseases. I hope you'll appreciate that research really has delivered some of the major advances in care that we've seen in the last 10 years in particular, 20 years, for some patients with some types of blood cancers. But that then leaves us with a major challenge for the other types of blood cancers with unmet needs. I hope you'll also appreciate that there really is enormous potential for the development of new therapies based on what we've learned over the last 10, 20, maybe 30 years about the biology of cancers of the blood. But equally, I hope you'll also appreciate that this doesn't happen by accident, that it requires commitment, a lot of time, fantastic teamwork, and what I'll call unwavering support to bring it to fruition. So, in closing, I want to make some acknowledgements and thank yous. The work I presented today, I've contributed to, but it's certainly not all my work. Over 100 researchers at WEHI in the last 5 to 10 years have contributed to this. Don will tell you later how many people contributed to his outstanding teamwork on the cytokine GCSF. I want to thank the clinical team at the Royal Melbourne Hospital and also at the Peter MacCallum for working hand in hand with us to test these new drugs, as well as the drug development team at the pharmaceutical companies Abbott and Genentech. Obviously, all the researchers at WeHi are absolutely indebted to those who've donated money to WeHi over the years to support our researchers, as well as to philanthropic groups such as Cancer Council Victoria, Leukemia Foundation and the Australian Cancer Research Foundation for providing grants to support their research. And most importantly, I really want to thank the patients and carers of patients with blood cancers, not just the patients that I've had the privilege of caring for, but for everyone out there. You're the reason why we do the research. We recognise that current treatments aren't as satisfactory as they need to be. We're absolutely committed to improving the lot of people that come after you. We're inspired by the courage that you need to show, and that stirs us on whenever we run into problems in the laboratory and things don't seem to be going right. All I need to do is go back over and see my patients to get a renewed sense of what's important in life and what needs to be done. So thank you for your attention.
You can sort of see why we get inspired when we've got clinicians like Andrew that come over and work with us and really relay to us firsthand the challenges that a lot of you have faced in tackling blood cell cancers. Thank you so much, Andrew. One of the things we like to do in these public lectures is to have an opportunity for you to meet a broader range of people working in the area and obviously to begin a conversation. So it's a privilege to introduce the panel members. Andrew's going to chair the panel. I'd like to introduce Professor Don Metcalf, whose work we've spoken about. Don is the Carden Fellow of the Cancer Council of Victoria, was for a long time the head of the Cancer Research Unit in the Division of Cancer and Haematology. Don? I'd like to introduce two of our wonderful young clinician researchers, Dr. Kylie Mason. Kylie is a clinician scientist working both in the Cancer and Haematology Division and as a haematologist within the Department of Clinical Haematology at RMH. And she's been part of that wonderful team that's developed those anti-cell survival drugs. Kylie, thank you. Dr. Ashley Nung is also a clinician scientist in the same divisions and has a similar appointment at the hospital and I think completing his PhD at the moment.
That is the plan.
That is the plan. You can see within the Institute, Andrew talked about teams. One of the really exciting things about working at the Institute is having teams that really span several generations. I'm sure if my successor is here in 20 or 30 years' time, you can bet Don will still be alive. He'll be nearly 100. But you can also bet that people like Kylie and Ashley will be the ones standing up here talking to you and celebrating the advances we've made. It is really one of the unique things about the Institute is having people of great experience working with people of great enthusiasm and talent. That's really special. And finally, somebody else who's very special and who has worked here for a little bit of the time, but her main job is a research nurse at Austin Health. She's a registered nurse with 20 years of experience caring for patients. She's a wonderful advocate for the needs of patients and has a wealth of experience. Rosie Hoyt.
So we welcome your questions. I, for one, certainly have done enough talking.
Yes. Just up the back there.
Is that me?
Yes, please.
Oh, okay. I've got a question about, I've heard a news item that they're now trialling giving people a lot of little bursts of chemotherapy more frequently. Is that going to become mainstream? Has it worked out to be better for the patients and that less toxic and less of a disruption to their lifestyle, or more of a disruption because they have to be treated more often? You know, has it worked out to be better or worse?
Sure. Well, one of the things about clinical research over the last 20 years is that we've been trying to refine how we use the drugs we've got available to us, and that certainly is one of the refinements that we've gone through. Kylie or Ashley, do you want to talk about that? Particularly in lymphoma, we're now compressing the treatment that we give for some of the aggressive lymphomas to try to get it over and done within 12 weeks rather than 6 months. That does mean the treatment's a bit more intensive and a little more toxic at the time. Have you guys got a perspective on that you'd like to share?
I think for all types of cancer treatments, we are aiming to try and maximise the effectiveness of it in our patients. And what Andrew didn't mention was that the first clinical trials in cancer were actually developed in children with leukemia, just down the road at the Children's Hospital. And it was Jonathan Colgan who made a decision and had a hypothesis, had an idea of how to treat leukemias in children, which was actually a fatal illness. And to the surprise and to a lot of resistance at the time, first treatments of children with types of chemotherapy agents actually led to their increase in survival. And this way of testing out these treatments in a randomised controlled trial has been the standard of therapy and standard of care in assessing new treatments and current treatments and how we approach treatments with patients with cancer now. So to answer your question, that approach of treating patients is going to be tested against our standard therapy, and we're going to be looking at how our patients respond. And if that's a significant benefit, then we'll take that forward, and that will become our next standard therapy.
It's certainly true that for some lymphomas, if you can get the chemo in shorter bursts, that seems to work better, but it's not true for all lymphomas. So as Ashley said, it needs to be tested. The other thing is that the benefits of the new treatment seem to be much greater than the benefits we're getting from just refining how we use the current drugs. So that's another reason why we're putting a lot of stock in the development of brand new therapies, not just improving the ones we've got. Yes.
Have you any idea how long, with myeloma, how long it takes for it to develop? And once it has developed to say 20 to 30%, how quickly it moves on from that? You know, does it just sit there for a while and you not know how quickly it will take off?
Okay, for those who may not have heard the question, it's about the disease multiple myeloma, and if you detect it with it at a certain stage, and 20 to 30% is often a relatively early stage, how long has it been there for, and how long might it stay there before it causes major problems? Is that a fair summary of your question? Kylie, do you want to?
So I think it's difficult to answer any question about any specific cancer because each cancer and also each individual is very, very different. So whilst one person might see 20-30% myeloma as compared to another person with 20 to 30% myeloma, those two people might have varying factors some that we might know and some that we might not. That will change the progress of their disease over time. Various genetic abnormalities that we find within the myeloma cells, but also the response to treatment, whether some people respond better to treatment, be that their cancer cells respond better, or they have toxicity that might lead to them stopping the treatment early or sooner than somebody else, for example. So it's very, very difficult to predict how one individual might go or how long their cancer might progress for or how well they might respond to chemotherapy. And myeloma in particular is a difficult disease. In some people we know that they have a prodrome to myeloma or a period where they have a very indolent disease for many, many years. In other people, we detect the myeloma when it's already a full-blown myeloma, and there's no way to really tell if those people have just had a more sudden progression to myeloma or in fact have had this prodrome that we've just never detected because they've never been unwell with it. So it's a very difficult question to answer in that there's a lot of variety between individual patients, and really we don't know enough about a lot of these diseases to be able to give an exact answer as to how long any of the diseases, be they myeloma, lymphoma or leukemia, have been there in any one individual person.
And just to add that, I mean, we don't think these happen overnight. One of the points I tried to make earlier is it takes some time for all of these cancers to develop, and the pace of change in an individual person will vary, just as Kylie said. Other questions?
Can you explain why early diagnosis of these types of cancers isn't an advantage?
Okay, so the question is, why isn't early detection of cancer necessarily an advantage? Great question, and I think the answer to it really lies in the difference between blood cancers and the cancers where we know that early detection does make a difference. Typically, those are cancers that start in a very specific organ in the body that are localised, and that there's an opportunity for cure through surgery or surgery through radiation, just to remove the whole thing. One of the problems with the blood is it's throughout us, and all the cancers that arise from the blood potentially are contaminated with, if you like, through the bloodstream at a very early stage in the development of the blood cancer. So unlike some cancers where how well you do might be determined has the cancer spread at the time of diagnosis, or how big the cancer is at diagnosis, for the blood cancers, generally speaking, it's uncommon if they're only present in one spot at diagnosis. Generally, we would assume that they're present in multiple areas, perhaps throughout the body. And so picking them up early doesn't change how we do a treatment so much, because we're going to need to treat the whole body typically with chemotherapy. Does that go some way to answering your question? Okay. Yes.
We've had some examples about how there can be a long time between initial discoveries in sort of basic research and then applications in a clinical setting. So I was wondering if the panel would like to say comment on the ways in which you measure your progress and know that you're on the right track, and how you, in those really extended timeframes.
Okay, so the perfect person to answer this question is Professor Metcalf. But for those who may not have heard, given that it may be a long way before you can really test how well you're going in terms of helping people, what are the stepping stones along the way that you can measure your progress? How do you know you're heading in the right direction, not wasting your time and people's money? Dom?
Well, you don't. Oh. But there are stepping stones. So, in talking about the colony stimulating factors, the first 10 years was spent trying to convince yourself that it wasn't just an artefact, that you weren't just picking up a contaminant in the water or in the medium that the cells were growing in. And it was a great relief to find a gene that actually coded for colony stimulating factors, because then you knew it was real. So that was progress. Doing a horrible artefact that lasted for a decade, now we knew there was a real thing. The next thing is, can you test your new discovery in an animal? Does it work? Does the animal ears drop off? Nothing happens. And so it's that. And the last hurdle is, will it work in a patient, and will it be a benefit to a patient? And sometimes you might have to wait another 30 years before you're sure about that answer. Some of the newer drugs that Andrew was talking about are doing a marvellous job today, but what's going to happen in 5 years' time, we don't know. Will the patient just redevelop the disease? And it's a very tough job with anti-cancer treatments, because you do have to wait 5 years, 10 years, 20 years before you think, well, no, it didn't work. Back to square one. So what you need are organisations that are prepared to support you year in, year out. And you try always to have side experiments going that are shorter term and are advancing knowledge. And you can at least say, well, I did that this year, I still haven't got to that step, but I've done so much. It's difficult. You can say that about any research. As we sit here, we have no idea what's going to happen tomorrow. That's half the fun of doing research. And you would say, yeah, but it's my money, isn't it? And it's true. It is our money we're spending, and we can't guarantee what we're going to discover. on average, over 10 years, we actually do what we promised to do 10 years ago.
So Don,
they're long-term.
So Don, you haven't mentioned publishing papers. You've published 900 thereof. Are they important? I mean, do they help tell us how we're going, or do they help disseminate the knowledge so other people can check whether what we're doing is sensible?
Yes, surprisingly to a lot of you, I think scientists write much more literature, wordage, than a novelist. Much more. That's our luck. And one of the things that we write are reports on what we've been doing, scientific papers. That does tell other people in the world what step in knowledge we've got to. It allows them to check to see if we've made a mistake or just plain told lies. So that happens. But as a group, scientists feel a very strong social responsibility to account for the money they've spent, which is the public's money. And the public are incredibly generous. Even if you forget the government one, it goes through our taxes. And I think we all feel obliged to count what we try to do, successfully do. So it's checks and balances.
Second one in, please.
It's my husband competing. With the clinical trials, I was just wondering how long they actually, what the length of them is. You actually may discover a new treatment. Do you actually look, have those same people been tested down the track sort of 5-10 years after they've had the treatment? Because whilst I'm sure anyone that's received some of the newer drugs is really grateful, I'm really grateful for it, we all have these side effects from the treatment, and I just wondered whether you continued looking at those people within that first clinical trials.
Sure. I mean, the short answer is on these clinical trials, we'll be following these people for as long as they are available to be followed. Rosie's got a lot of experience in following people on clinical trials of some drugs that go forever, such as the drug imatinib or Gleevec for CML. Typically, we will just keep that going as long as it's working, and so monitoring people long term is crucial. Rosie, do you want to talk about monitoring people on clinical trials and your perspectives on that?
Sometimes if the trials are sponsored by pharmaceutical companies, there's only a certain amount of information that they want. They find out that the people have responded very well and they haven't, and they collect that information. But what I suppose we're always very interested in is how people are going on a longer term basis. Sometimes the pharmaceutical companies want that long-term data, but sometimes they may not want it, or they may think in a few years' time, oh, we should have asked, and then they come back to us and say, well, what's happened to those people? I find it, I think, really rewarding to continue to follow people to find out how they're going with if they're having longer-term sort of side effects that we never thought that they would develop, and then being able to report that back to increase our body of knowledge, so that when we are educating people who are newly diagnosed, we can say, well, we've had people who are on this drug now for say 10 years and they're all doing really well, they haven't got any sort of unusual effects from it, or perhaps some have, or it's affected maybe their fertility that we didn't think it would, or it's affected other sort of aspects of their life that we weren't quite sure if it would or not. So I think it's very important when we're offering people clinical trials to be able to, as much as we can, to continue to follow them for a long period of time, to be able to collect that additional information, so that when you come back and see a newly diagnosed person, you can give them as much information as possible about what's going to happen now, or what's going to happen in 6 months, but what also might happen in 5 years' time, so that they can. Sometimes people don't want to know what's going to happen in 5 years' time because that's too far ahead for them to look. They're only worried about what's going to happen today or tomorrow. But other people, depending on who you are, everyone's different. The way that they want to, the way they look at things. It's nice to have that information.
Yeah, I mean, clinical trials are just part of clinical care, and at various times for some people they're the most important part of clinical care, but by and large they're only one aspect of it. Oh, that's a great question. We'll ask your hubby now.
My question, or as a comment, and I'm probably looking for some comments back, as an observer of 3 very close people to me who've had cancer, are you people seeing that with the rising numbers that the health system itself actually provides the care that's necessary to get the best result? My observation would be our present setup is that the GPs is where you go first and you get referred on down. But somehow or other, I don't know whether it's through funding or whatever, there always has to be the link that goes back to the GP. But my observations are that a lot of the time the GPs are just dead scared once it's moved on from them to be involved in any comment or diagnosis of any sort about the side effects and all of the things that are happening. And do we need to perhaps change the way that's done or not? Because I think it's creating a lot of duplication back and forth and probably creating a lot of uncertainty with the patients as they ping-pong back between sort of the ailment and the treatment of the disease.
Yeah, look, I'll take that one if I can. There's no question that in Australia generally we have very good access to very good treatment, but it's not perfect and it's under huge strain. And that's not just the case for blood cancers, it's for all cancers, it's also for people with life-threatening heart disease, strokes, etc., etc. And perhaps the part of the health system that's under greatest pressure is the general practitioners. And a lot of work is now going into trying to heal that gap, but it won't be solved for another 5-10 years at least, when this large number of new doctors comes through the system and gradually gets out into GP land to take up the cudgels where current GPs are just snowed under. One of the reasons GPs generally like to become disengaged early on is because we've talked today about blood cancers, I've talked about they're increasingly common and things like that, but the average GP will see one person with acute leukemia once a decade. So they don't have the experience necessarily readily at hand. That's at the hospital. For the more common blood cancers like lymphoma, GPs will have more experience, and I think in future GPs are going to be more involved in the long-term follow-up of our patients because the hospitals cannot cope with all the long-term follow-up that's going to be required as we increase the percentage of people who have survived. So that is one area of healthcare that needs attention, but I think the solutions are a little way away until we solve some workforce issues. But the government's aware and is trying to do something about that. Other questions? Yes.
Have many species of animals blood orders? And if so, are they used in research?
Don, do you want to address that? Blood cancers occur in many different species.
All species get leukemia and blood cancers. And there are some mice where every member of that particular branded mouse will get leukemia, just spontaneous. Poultry get leukemia. The mass farmed poultry have dozens of cases of leukemia. You probably had eggs from lots of them. Usually around with a paralysed leg or a paralysed wing has in fact got leukemia. It's a very common disease in white people. Cats get leukemia, dogs get leukemia. The thing that put everyone off was that in all animals, just about, it's obvious that leukemia is caused by a virus, one or other type of virus. So everyone in the 50s believed with their heart that humans would prove to have leukemias that were caused by viruses. And to this date, they haven't been found, except for one type which causes leukemia in southern Japan and in the West Indies. A relative, not close, but a relative of the AIDS virus. Other than that, apparently we're the exception. And still we say, hang on a minute, if cattle have viruses, if chooks have viruses, if mice have viruses, we must have them. They haven't been found yet.
And we're still looking. We've got time for just a couple more questions. Yeah.
You mentioned the development of the drug type BCL2 and that there's research on second generation drugs. Are any of those approaching clinical trials yet, or are they still
Well, companies around the world are working very hard on that. I don't know the answer to your question. My hope is that they're not too far away. The only trials that are happening at the present time are with the drug that I talked about. We're still pretty early in its development, and I think we're that the priority at the moment really should be to just see in which type of blood cancers, or in fact other cancers, this drug has potential to make a difference for people, and then to see how we can combine it with other drugs. And while we're taking care of that, I'm very confident that newer, perhaps better drugs will come along. Yes.
You mentioned in your talk that radiation causes cancer. By the same token that we have lymphoma, we usually give them the course of radiation, which is the same exposure. Marie Curie developed a lymphoid problem with her radiation, and yet that radiation was so severe that you can't even read her diaries now. And yet she produced 3 children, all of whom were genius. Should we stop radiating our humans?
Yes. So we're far more, because of the experiences that you've described, I mean, we're far more aware of how better to use radiation, and it's like most things, use it to its best advantage, hopefully in circumstances with the least side effects. Kylie, do you want to touch on that one?
So, as Andrew was saying, it's about whether it be radiation or chemotherapy, understanding best how to use a particular therapy to minimise or to balance or weigh up the benefit with the toxicity. And I think a lot of our treatments are designed, and whether that's a single drug or a single radiation treatment, or it's a combination, as we were talking about earlier, of multiple drugs together, or drugs and radiation treatment, to balance that benefit of therapy or cure, or the aim of cure, with the toxicity. And Marie Curie was obviously so influential with radiation. We learned so much from her experiments that we now know, and from that time onwards, we know so much more about radiation, that we know what are safer doses. And that's progressed a long time, even since the 1980s, for example, when they gave very high doses of radiation to children with leukemia, in the thought that that was the best therapy at the time for them. And they're now discovering that these children are developing tumours of the brain, for example, 20, 30 years later. Something that they didn't know and didn't intend back in the 1970s and 1980s, but is having a profound effect now. So the long-term follow-up that Rosie was talking about earlier is particularly important in monitoring all therapies, not just radiation therapy, for example, but all therapies, to feed back in the long term this balance between therapeutic possibility of cure and toxicity in the longer term.
And I'm right in thinking we use significantly less radiation in fewer patients with acute lymphoblastic leukemia in 2010.
So now in 2010, radiation treatment is no longer a standard treatment for the majority of children who have acute leukemia, nor is it a standard treatment for a lot of disorders that would have been back in the 70s or 80s, for example. And it's used in a much more specific and targeted way.
All right, now I'd like to hand back to Dr. Hilton.
Thank you, Andrew. Thank you to our wonderful panel. I'd like to ask 2 people to stand up. David, could you stand up? And Sue O'Brien. David's from the Cancer Council of Victoria, and Sue O'Brien from the Leukemia Foundation, and they'll come into the tea room, no doubt. you want to talk to David or Sue about what their organisations are doing to help in prevention, treatment, diagnosis of cancer, I'm sure they would be delighted. Our head of fundraising is Deb Cutts, and again, if you'd like to talk to Deb about supporting the research that goes on here, of course, she'd be delighted to talk to you. Finally, I'd just like to thank all of you for joining us on a wet winter's day at the Walton Lifelong Institute. It's been a real pleasure for us to be able to share some of the research that we're doing with you, so thank you very much. The tea room is just out the doors to the right, so please join us.